Let's look at the embryonic development during week 4, day 22 to 28. The embryo is in transition. It is in the process of gastrulation and neurulation. And this neurulation has driven the embryo to fold cranially, caudally, and laterally. So this embryo at this point kind of looks like a little messed up comma shape. There's oropharyngeal membrane and cloacal membrane marking the cranial and caudal ends of the embryo. In front of the cloacal membrane, there's still primitive streak present. It's still the same size as the middle of week 3. It just looks small because relatively speaking, the rest of the embryo has grown and elongated quite significantly because of all the tissues that are being generated from the primitive streak. From the primitive node of the streak, we have the nodal cord that has extended up towards the oropharyngeal membrane in the mesodermal layer. This nodal cord appears curved as well because it is inside the folding embryo. The ectoderm just dorsal to the nodal cord has thickened into neuroectoderm and is in the process of neurulation. Both the cranial and caudal ends of the neural plates are still open and continuous with the regular ectoderm, but in the mid-region of the embryo, the neural tube has sunken under and has pinched off of the ectoderm at this point. Coloring in the ectoderm in this dark blue, we can really see where the skin or the ectoderm is continuous in the mid-region of the embryo, and the neuroectoderm in lighter blue in the cranial and caudal regions that are still continuous with the skin will need to sink under and pinch off of the skin to complete the neurulation. And here's that partial neural tube that has sunken under the skin and pinched off. Okay, let's remove that ectodermal and neuroectodermal coloring to see what's going on underneath the surface a little more clearly. Along the length of the neural plate, the specialized cells at the margin between the neural plate and the ectoderm are emerging, which are the neural crest cells, the fourth germ layer. Where the neural tube has pinched off of the skin, the neural crests are in the process of migration on either side of the formed neural tube. And in the cranial and caudal regions, as the neural tube continues to zip up cranially and caudally, the neural crests will continue to form and migrate laterally as well. Remember that neural crests are forming through the process of EMT, the epithelial to mesenchymal transition. On either side of the developing neural tube and the neural crests, from the primitive streak, we have a pair of mesodermal streaks that are migrating cranially. These are the paraxial mesoderm that ball up starting from the mid-region of the embryo in regular intervals in caudal direction. These balled up proximal mesoderms are called the somites. Lateral to developing somites, coming from the primitive streak, we have a pair of intermediate mesoderms that give rise to the urogenital ridge, the primordia of the kidneys and gonads. And in the lateralmost edge of the embryo, going all the way even cranial to the oropharyngeal membrane, we have the last set of mesodermal tissues, the lateral plate mesoderm. Within that lateral plate mesoderm, we have a tunnel that runs through it, and this is the intraembryonic coelom. So we have the lateral plate mesoderm here that has split into 2 layers. When this space formed, the dorsal layer is called the parietal layer, the ventral side is called the visceral layer, and the intraembryonic coelom has formed in between these 2 layers. This horseshoe-shaped fluid-filled cavity will compartmentalize and become the pleural cavity, pericardial cavity, and peritoneal cavity. Where's the endoderm in this story? It is still there. It is ventral to all of those mesodermal layer. And if you may remember, the endoderm is continuous with the yolk sac, which is still present right here. And speaking of, the ectoderm of the trilaminar germ disc itself is also continuous with the membrane of the amniotic cavity. So as the trilaminar germ disc itself is folding cranially, caudally, and laterally, these 2 bubbles, if you will, the yolk sac and the amnion, are also morphing along. Some other extraembryonic structures, just for the reference, are still present. Towards the cloacal membrane, we still have the connecting stalk, aka the umbilical cord, which is comprised of the extraembryonic mesoderm, which coats the outside of the yolk sac and the amniotic membrane. So these structures are still there, they're not going anywhere. But we'll disappear them for now so we can focus our attention on the embryo proper development. development. Let's take a cross-section through the middle of the embryo and see what that looks like. Here's the ectoderm that has curved quite a bit due to the lateral folding. The neural tube has formed and pinched off of that ectoderm, and ventral to that we have the nodal cord, and on either side of that neural tube we have the neural crest cell population. Lateral to all of these we have the mesodermal layers in 3 paired streaks. And ventrally and getting incorporated into this laterally folding embryo is the endoderm. We're still kind of trilaminar germ disc with ectoderm, mesoderm, and endoderm still present, but this is no longer a regular flat ravioli. This is a ravioli with anatomy that is also folding cranially, caudally, and laterally. Neural tube, neural crest, and here are the extraembryonic structures for reference. Again, during the 4th week, the gastrulation and neurulation will continue to ensue, driving the growth and curvature of the embryo even further. The oropharyngeal membrane, which used to be at the cranial end of the embryo, has now been brought ventrally and caudally, and likewise the cloacal membrane, which used to be on the caudal aspect of the embryo, is now brought forward ventrally and cranially. There's still primitive streak, but it is starting to slow down a little bit. In fact, it is starting to regress. The neurulation has progressed to the point where the cranial portion of the neural tube has now completely folded and sank under and pinched off of the ectoderm. The caudalmost portion of the primitive streak is still continuous with the regular ectoderm, but soon it too will complete its neurulation and pinch off of the skin by about day 28. With the progressing neurulation, there's of course the neural crest cell populations that are emerging as well, and immediately lateral to the formed neural tube and neural crests, we have the paraxial mesoderms that are forming the somites. Lateral to that, we have the intermediate mesoderm, and lateral to that, we have now quite complex lateral plate mesoderm that has split into ventral and lateral sheets that are together folding ventrally. It's hard to actually draw these 2 layers out in this lateral view, but what I will draw is the intraembryonic coelom in between these 2 layers of the lateral plate mesoderm for the time being. With this exaggerated folding, the double bubble situation is evolving as well. Here's the endoderm that is part of the trilaminar germ disc that is still continuous with the yolk sac, but the shape of the yolk sac is starting to change a little bit. The amniotic membrane is expanding, still continuous with the ectoderm. So here's the yolk sac, and this portion of the yolk sac with the endodermal lining that's been incorporated into the cranial part of the folding embryo is called the foregut. This middle part is called the midgut, and this caudal part under the tail of the embryo is called the hindgut, because what used to be a spherical balloon-like yolk sac is eventually going to give rise to, you guessed it, our gut tube. And in between the part of the yolk sac that is now becoming a part of the embryo and the part that is kind of getting squeezed outside of the embryo, which we'll still call the yolk sac, there's this narrowing channel called the vitelline duct. Here's the amniotic cavity that's expanding. So this is about mid-week 4, day 25. With this continued folding, expanding, and growing, let's see what the embryo looks like by the end of week 4 at day 28. The embryo at this point has completely curled ventrally so that the entire exterior of the embryo is covered by the ectoderm. And still continuous with that ectoderm, we have the amniotic membrane that is forming a complete sphere around the developing embryo. The amniotic membrane has in fact draped over the umbilical cord and forms its outermost layer. Let's remove that amniotic cavity for now. By the end of week 4, the caudalmost neural plate that was still continuous with the skin has now folded under and pinched off of the skin. So we now have completely skin-covered embryo that looks like a comma. So the neurulation is complete. Furthermore, the primitive streak, the tissue factory, has completely degenerated by the end of week 4. So there's no more tissue primordia that is being formed. The oropharyngeal membrane and the cloacal membrane are positioned ventrally now. The yolk sac that used to be a spherical bubble has now been incorporated into this folded embryo, so that we have the part of the yolk sac that is incorporated into the embryo in a tube-like shape, and a tiny part that's squeezed outside of the body, but still connected to each other by a thin channel called the vitelline duct. The cranial portion of the tube is called the foregut. This middle part that is still continuous with the yolk sac is called the midgut, and this caudal part is the hindgut. The paired paraxial mesoderm layers are still there, doing their thing by forming somites. We're only seeing the left side of the embryo right now. There should be another set of these somites on the other side of the screen. Lateral and now slightly ventral to the somites, we have the intermediate mesoderm forming the urogenital ridge. And attempting to draw the parietal lateral plate mesoderm in this purple color, it is just under the ectoderm or the skin. It is forming the inner aspect of the lateral and ventral body wall, and attempting to draw the visceral lateral plate mesoderm, coating the outside of the endoderm lined tube structures in the body. So it follows all the gut tube contours. And another important thing about visceral lateral plate mesoderm is that it's from this layer that the heart tube is forming and protruding out into this little bulge that has formed just cranial to the umbilical cord. And in between this purple and red layer of lateral plate mesoderm, in green color, there would be the intraembryonic body cavity that has also transformed into this curved shape due to the elaborate embryo folding. So now this completely folded embryo is surrounded entirely by the amnion. And to revisit all the extraembryonic structures, we still have that extraembryonic mesoderm coating the outside of the extraembryonic structures, the yolk sac, and the outside of the amniotic membrane. And with this folding, the connecting stalk or the umbilical cord that used to be at the caudal-most end of the embryo has also been folded upwards or cranially. And it fuses with the extraembryonic mesoderm that covers the yolk sac and the amnion and forms the umbilical cord proper. Once again, let's remove all of these extraembryonic structures except for the yolk sac and a stump of the umbilical cord, and take a look at the cross-section of this 4th week embryo just caudal to the umbilical cord. The embryo is now completely covered by the ectoderm, which is a good thing because ectoderm becomes the skin, and we certainly want to be covered by the skin on the outside. The neural tube has sunken under and pinched completely off of the skin thanks to the nodal cord induction. Along with the neural tube, we've also formed the neural crest cell population. Lateral to the neural tube and neural crests, we have the paraxial mesoderm. Laterally and now ventrally, due to lateral folding, we have the intermediate mesoderm. And now, the 2 layers of the lateral plate mesoderms have gone a significant amount of transformation due to the lateral folding. The parietal LPM has become curved just under the ectodermal layer. Likewise, the visceral LPM have folded ventrally and medially, surrounding the outside of the gut tube, derived from the yolk sac. And between the visceral and parietal layers of the LPM, we have the intraembryonic cavity. Soon thereafter, the gut tube will end up pushing out into this intraembryonic cavity to develop further. So the visceral LPM accommodates this by pushing out along with the gut tube, helping it narrow down into a tube shape, and then suspending that gut tube while anchoring it to the back body wall by the right and left side fusing together to form a sheet of connective tissue called a mesentery. So at this point, the lateral and anterior body wall of our body is comprised of the parietal LPM and the ectoderm, while the wall of the gut tube is comprised of the visceral LPM and the endoderm.